Yeah. Oh, I'm now disabled from the sharing.
1 second. Maybe I can only have a maximum number of. I will try. Now?
Yeah, it works now.
Oh.
Is it okay now?
Yes.
Should be okay, great. Okay, so welcome everyone and good afternoon to everyone who is following us today. And we met again for the Tropical Ecology Online seminar, which is hosted by the Society for Tropical Ecology. And the society is a very active society which organizes the annual international conference. We hope to have the next one in 2022 in person, hopefully. And the society also organizes the journal Ecotropica, where you can get the peer-reviewed manuscript published for no costs. And the society is also giving the grants and awards to travel and do follow-up research. And we are also giving the award to the student good research of the students as the Marian Award. And finally, the society has got a very active student group where the students can join and discuss the actual topics and get the insights on what is going on in the tropical ecology. And today, we are meeting as every month for the seminar where we are discussing a topic, selected topic by a senior researcher and one or two younger researchers which are showing the, like a case study. So today I would like to welcome Knud Jønsson and Kasun Bodawatta, who are both based at the University of Copenhagen in Denmark. And we will be discussing the topic of barriers, which means the mountains and the barriers which might be preventing the dispersal of species, and also the diversity along the gradients of various tropical systems. So while Kasun will be focusing more on the diversity and formation of New Guinean avian diversity across the mountains, Kasun will focus more on the avian gut microbiome in these birds. And I think this is all from me for the beginning, and I would like to give the space to Knud now for 30 minutes to present his research. Thank you and welcome.
Right, I wonder if you need to, are you unsharing first?
Yes,
or?
Exactly. I have to stop sharing.
Yes, then I'll
Your turn now.
try and get in there. All right. Okay, thank you very much, Katka. It's great to be here. My name is Knud Jønsson. I'm an associate professor at the Natural History Museum of Denmark, which is also a part of the University of Copenhagen, also in Denmark. I'll be talking today about the formation of New Guinean avian diversity across barriers and along elevational gradients. And this opener here pretty much shows what I do. It's all about New Guinea and some of the surrounding islands, and it's a lot about birds. So, oh, how do I need to get down there? All right, so here's an island, not just any island, it's the largest tropical island in the world. I guess a lot of people, at least from Bodawatta, would know this island. It's got enigmatic, at least, biodiversity on it. It's got birds of paradise, tree kangaroos, and a bunch of other really cool things. It is, in itself an island system, at least a montane island system, in that it's dominated by a central range, a very tall montane range that cuts through the island, and it also has a number of little outlying ranges in the perimeter of the island. So that creates its own little, or not little, own large archipelago of mountains and lowlands. It didn't always look as it does today. So this is just a very brief overview of how New Guinea formed back in time. So the top two maps are from 10 million years ago and 5 million years ago. and so the island is sitting on this northern rim of the Australian plate. 10 million years ago, you can see these white and red bricks here. Those are representing calcareous platforms. So very low-lying potential land that may or may not have been above sea level. And then there's a bunch of scattered islands, the green little blurbs out here. And that is pretty much the pre-New Guinea. What later then, some 5 million years ago. Can you see my cursor, by the way, when I move it? Yeah. So 5 million years ago, we're starting to get some highland regions here, the orange patches. And we have this whole island arc going out here, which is today the Bismarcks and the Solomon Islands and all the way out to Fiji, that then clashes into this land here. And we start to form mountains. And then today, the lower map here, we have some central mountain regions that are quite old. And we have some lowland regions and some outlying mountain ranges that are relatively young. So here's a little bit of a breakdown. I like maps a lot, so there'll be a lot of maps today. Here's another depiction of New Guinea. Red is montane areas, and even the lowlands can be broken into some specific lowland basins that formed at different times. The main message here is New Guinea is about 800,000 square kilometers, and the majority of that land, some more than 500,000 square kilometers, are lowland regions. The highlands are smaller, but still relatively extensive, and so more than 250,000 square kilometers are above 500 meters, and as you can see, 200,000 of those square kilometers consist of the central range. Then these outlying ranges vary in size. So we have the Huon Mountains down here that are some 14,000 square kilometers. And then we have the tiny, tiny Cyclop mountains that only consist of 160 square kilometers. So there's a lot of different montane islands in all sorts of sizes across the island. So what I'll be talking about here today, it was a little bit of a lie when I said I'd talk about avian diversity, because I'll only talk about passerine bird diversity. And this work will then talk about the 336 passerine bird species that breed on a regular basis on the main island of New Guinea. So first off, I'm not talking about vagrants, and I'm not talking about introduced species. So first off, we constructed a molecular phylogeny or molecular tree of these 336 species, and that's based on a simple approach of sourcing material from GenBank, 3 nuclear, 3 mitochondrial genes. But then some 40 species were not available, so we've sequenced those as well to make sure that we have absolutely all the species, all the passerine species of New Guinea, and that's the tree you see here on the right. All right, let me walk you through some of the patterns that we found and some of the interpretations of what those patterns mean. So first off, we broke New Guinea into these elevational bands, so from 0 to 100 meters, from 100 to 200 meters, and so forth. And what we see here is that we have by far the highest species richness in the lowlands. So if we take the top curve here with the full circles, then that represents all passerine birds. Highest species richness in the lowlands, and then it drops pretty much, maybe it's plateauing in the beginning, and then it drops as you move up to high elevations. If we then break passerines down into non-endemics and endemics, so non-endemics are those that also occur outside of New Guinea, then we see a very similar pattern, that species richness simply drops as you move upwards. But the pattern of the endemic species is somewhat different in that it actually increases as you move up to mid elevations, and only then around 2,000 meters does the richness start to decrease. If we then look at smaller spatial scales, so grid cells that are 11 by 11 kilometers, the pattern changes a wee bit. Oh, so I should say in the first one, it's probably obvious that because we have more area, we also have more species. But now we're looking at smaller spatial scales. So we're looking at grid cells that are 11 by 11 kilometers, and the pattern changes a little bit in that now we actually see the highest species richness at mid elevation, so probably topping around 1,200 meters above sea level. And again, that is driven by the endemic species, the light gray here, and non-endemics pretty much just decline in richness as you move up. We can depict that in a map, and that also shows that the species richness is high. So we have the central range here with low species richness, but just on both on the north slope and the south slope, and the south slope down here in the Owen Stanleys is where we actually have the highest species richness. Now, an interesting observation is in the mean range overlap of species. So there's a high overlap of species ranges in the lowlands, meaning that if you occur in the lowlands, then you're very likely to coexist with other lowland species and have ranges that are roughly or very similar. Then it drops. So the species range overlap is very small at mid elevations, probably because we have highland species coming down, lowland species coming up, and they just overlap a tiny bit. And then it increases again. So if you're a highland species, you're very likely to overlap with the ranges of other highland species. And finally, we're just taking a look at the mean range size of species. And again, perhaps not surprisingly, the ranges of lowland species are the largest, and range sizes simply drop as you move up with a slight plateauing again at mid elevations and then a drop. So highland species have the smallest ranges, probably not a surprise either, given that there's very little area up at high elevations. So the take home of all this is that there are more species in the lowlands because there's more area. Mid elevation peak in richness at local spatial scales. Some of the lowland species or the species richness can be explained in light of allospecies. So in some cases, we have species that are differentiated, very closely related species that replace each other geographically. So in the lowlands, you get many species, but if you look at a particular grid cell, you actually get relatively fewer species. We have a continuous drop in richness with elevation for non-endemics, but not for endemics. And the proportion of endemics increases with elevation. Oh, and mid-elevation richness peak coincides with low range overlap. So just to hammer home this point about endemic species, this is a plot of the proportion of endemic species as you move from the lowlands to the highlands. And you can see that proportion goes up quite markedly. And once we get up to the highest elevations or mid elevations and upwards, we have avian communities that are dominated by endemic species. This is the same case at smaller spatial scales at grid cell level. And when we depict it on a map, again, we can see that the proportion of endemics is very high at the highest elevations. And now we're adding one more spatial scale to this. These are the stations along the Mount Wilhelm transect. And even here, for passerine birds, we can see that the proportion of endemic species increases as we move upwards. Now switching a little bit to ages of species. So how old are these species? And in the first one here, we're back to the elevational bands. And what we can see is that the mean species age is quite low in the lowlands. Then it climbs as we move up, and we have the graph here is when we exclude endemic families. So again, there's an increase here in species age, and that is driven by endemic species. It still goes up. We still have older species as we move upwards for all species, but it's particularly so for species that belong to endemic families. The pattern is similar at smaller spatial scales. Again, that translates into a map that looks like this. So we have older species in the highlands. And then one thing that's notable here as well is that we have a little bit of a divide. So these are the northern lowlands here, where species seem to be older than in the southern lowlands, and notably in this area here, the transfly, which is very close to Australia, which is just down here. And again, along the Mount Wilhelm transect, we find a similar pattern that species ages go up as you move from the lowlands to the highlands. And again, notably driven by endemic species. So here, what we find is that the proportion of endemics increase with elevation, species age increases with elevation, and species age increase is driven by endemics, notably by endemic families. And here's another little piece of evidence that shows that if we break the species into endemic family species, so these are representatives of families where all the species of those families only occur in New Guinea, berry peggers, ifrits, and some other berry peggers, two different kinds of berry peggers. And the endemic family species are quite a bit older than endemics and non-endemics, which are quite similar in age. And then just a little aside here, this is another depiction of the species richness, which is highest in the lowlands and also fairly high in both the central range and the outlying ranges. What we can see here is if we break species into certain age groups, so it's pretty much we're slicing the phylogeny and taking all the species that are younger than 3.5 million years and all those that are older than 3.5 million years, we actually find that the number of species in the lowlands that are younger than 3.5 million years is quite high, whereas we have quite few of these young species in the central range and the outlying ranges. However, the proportion of endemic species looks a bit different in that we have about half of the species of the endemic species are younger than 3.5 million years in the lowlands and the central range, but in the outlying ranges, we have a markedly higher proportion of endemic species that are relatively young. So the take home here is that we have many young species in the lowlands, we have few young species in the highlands, and the proportion of young endemic species is high in the outlying ranges. Many species are shared between the central range and the outlying ranges. And so in some, we have these young, widespread and non-endemic species in the lowlands. We have old endemic species in the highlands, and we have both old and young endemic species in the outlying ranges. The interpretation of all this is basically that species colonize through or form in the lowlands. Over time, species move up and specialize. In this case, they become endemics that only occur in New Guinea. We also have some morphological data of some of these birds that support the idea that these species actually are fairly specialized compared to the rest of New Guinean passerine birds. And also, there seems to be some exchange between the central range and the outlying ranges. So even though things move up, it's not a dead end. There is still some movement out to the outlying ranges. And this is one of the things that we've also been very interested in and wanted to look further into, and that spurred the tentative working title, Barriers and Gradients, because in this project or in this study here, we wanted to know how do populations differentiate across barriers, and do levels of differentiation differ between lowland species and highland species? So we randomly chose, no, we did not randomly choose anything. We went out and collected what we could and tried to put together a data set of a broad range of species from New Guinea that have populations both in the central range and in this case, the Huon Mountains. So this is what we have here. We the primary target area was Mount Wilhelm, and then we compared with populations from the Huon Mountains here. For the lowland species, we also included populations in between here from Wanang, and for some of the highland species, we included a third population from down here around Mount Scratchley. And so this is the selection of species that we can see here. So we have genomic data for 14 species of 2 to 3 populations. We have 12 de novo assembled genomes, and then we have 233 resequenced samples. So we were aiming to get a minimum of 5 individuals per population, and in some cases we had 10 individuals per population. So at least large enough numbers that we can do some sort of demographic analysis of what is actually going on in 2 different populations. So first of all, one of the things that is clear is that when we look at the elevation that these birds occur at, that is the minimum elevation that they occur at, the higher up in the mountains you are, the more differentiated are the 2 populations. So in terms of highest level of differentiation, Paramythia montium here is quite well differentiated between Mount Wilhelm and the Huon Mountains. And when we go to the lowlands, Melilestes, a honey eater, and Toxorhamphus, they're basically panmictic. So they just mix completely between the Mount Wilhelm lowlands and the Huon lowlands and the Wanang lowlands. Then we went on to look at the population sizes through time, and what we see here is a number of different things. So let's try and do it slowly here. So first of all, these are 3 examples of the species included in here. It's a lowland species that is essentially undifferentiated. It's a mid-elevation species that is relatively undifferentiated. We have a bunch of these mid-elevation species, and some of them are you know, to some extent differentiated, and some of them are pretty much panmictic as well. And then we have a highland species here with an FST of 0.09. So it's a relatively well differentiated between the 2 mountain regions. The orange shading here represents the Pleistocene time period. And what is immediately noticeable when you look at all 3 patterns is that they go up and down quite extensively, and most notably within the Pleistocene. Part of that explanation is probably that is a time period where there were enormous climatic changes, so cooling and warming, and that would mean the forest expanded and contracted. So probably as forests expanded and contracted, so did the distributions and ultimately the population sizes of these different species. The thick brown line here represents the de novo assembled genome. So that is the most reliable of the different curves. But what you can see is that we can break down individuals into the different populations that they were sampled from. So in this case here, blue is Huon, purple is Mount Wilhelm, and green is Wanang, that lowland area in between. And it is an undifferentiated, the populations are undifferentiated, and they pretty much follow the same population size trajectories through time. The little lines out here represent another method of calculating effective population sizes, and we're pretty content when we get values that are so similar here. For Melanocharis, it's pretty much the same thing. It goes up and then it increases and then decreases towards the present. And the trajectories do pretty much the same thing. In the final example here with Paramythia, we can actually see that the Huon population, the blue lines here, they decrease towards the present after an increase, whereas the central range populations, they seem to continue to increase or at least plateau up here. So we can pretty much follow that the different trajectories that these populations go through. On top of that, on top of calculating effective population sizes and sort of gauging the divergence times, we can also do it more explicitly, do more explicit demographic modeling. We used FastSimCol2, and with that, you can also calculate or estimate the effective number of migrants. So that's pretty cool. That means, if you trust it, that you can calculate how many individuals move between populations per generation. So just to try and break it down, I've broken it down into the high elevation species, the mid elevation species, and the lowland species. And what you can see here is that the less or the more differentiated populations are, the lower are the effective number of migrants. So they're pretty low up here, they're pretty high down here for the lowlands, and we get a bit of a mixed bag here in the middle for mid elevation species. Now, that's one thing. What I also find very interesting is that, so even though they're well differentiated in the highlands, there are still dispersal events between the populations. So that means, even though, yeah, they're well differentiated, there are still individuals going back and forth mixing in. So we don't have, Paramythia here is differentiated at species level comparatively to other species in New Guinea and the rest of the world, but still there are individuals getting across every now and then. Not many, but some. Another thing that is noticeable is that we have this idea that most individuals would disperse from the central range to the outlying ranges, and definitely in the majority of the cases, the values are higher from the central range to Huon than the opposite way. But there are cases where there are more individuals going the other way. And so the interpretation here is that even though they disperse in both directions, colonizers from Huon to the central range would meet very saturated communities in which it would be very difficult to establish, whereas the small populations and the small outlying ranges are more prone to extinction. And so more often, dispersers that arrive in the outlying ranges would have the ability to establish populations. And that could also explain why it is that we get some of these young endemic species in the outlying ranges. So the take home is that populations of high elevation species are more differentiated than populations of low elevation species. Dramatic changes in population sizes during the Pleistocene. Dispersal both from the central range to Huon and from Huon to the central range. And that ultimately leads you to start thinking about taxon cycles, because a lot of these things tie in nicely with the idea that species go through these phases of expansions and contractions. So just briefly, taxon cycles predict that the phases from a species from a source pool expand to other islands nearby that they colonize. After a while, they've colonized the whole island. They start in the perimeter, then move inwards. Then they differentiate and become different taxa or different species. Then we have the contraction stage where things die out on the smallest islands. And then in the final stages, we have relictualization, where we only have these differentiated species left on the largest islands and often at higher elevations. That's what E.O. Wilson came up with based on his ant work in the Pacific a while ago. So that is essentially what I think is happening in New Guinea as well, that things come in through the lowlands and then in some cases differentiate into these allospecies and then over time move upwards and become these relictual endemic species in the montane regions. But then with that additional little twist that some of the montane species still disperse out to other smaller montane areas on New Guinea. And that concludes my talk. I mean, basically, when I say, that's the classic, when I say I, that means we. there's a lot of people that contributed to this work, notably museum collections worldwide, and others, the students and postdocs, a lot of local collaborators in Papua New Guinea, and especially the Binatang Research Center contributed massively to this work. And of course, funding from the Carlsberg and the Willem Foundations. Thank you very much.
Thank you, Kasun. Sorry, thank you, Knud. That was wonderful. I am ahead of my thinking. And now I would like to give the space to Kasun to follow up with his research on the microbiome of these same birds, I believe. Thank you. And the questions will be coming soon after Kasun's talk.
Yeah. All right, should I start? I see. Hi, everyone. Thank you very much for inviting me, and especially Katrina Sam for inviting me to give this talk here at the Society for Tropical Ecology. And now I'm going to take you to a little bit finer scale than Knud's talk, where I study gut microbial symbionts of these tropical birds and try to understand what drives these associations in these diverse communities. So before I give the background or take you through the most of the results from my work, I would like to give you a small background on avian microbial associations. This is one of my favorite illustrations to show how entangled we are in a microbial world, and we don't have to look that far. When we look at our own mitochondria, we do see the importance of animal microbial associations to sustain life on Earth. And when we look at these animal microbial associations, they can be simple, where we have a one host and one microbial symbiont, like in the famous bobtail squid and Vibrio fischeri association. And then these microbial communities can also be highly complex and diverse, like what we see in our digestive tract. And these digestive tract microbial communities, they play multiple roles for their hosts, mainly on many digestion-related functions and immune-related functions, and also developmental and behavioral functions. And in many cases, hosts cannot survive without these functions. These microbes are playing for them. And while microbes playing these roles for the host, they also interact with one another, making it really complex to understand how these communities are assembled in these digestive tracts. And because of this important roles microbes played for their hosts, it's also important to understand the functions played by these microbes and also drivers of these microbial communities to fully understand the host biology and ecology, and in some cases, the evolution of the host. And so during my PhD, I looked at avian gut microbial communities and tried to understand multiple factors driving these complex associations. And I choose birds because they are phylogenetically diverse, and also they are globally distributed, and also they occupy multiple niches, providing a great phylogenetic and ecological diversity to investigate the factors governing these complex symbiont associations. And again, when I talk about birds, I mainly focused on the passeriforms, passerines, like Knud talked about. And these birds occupy around 60% of the extant bird diversity. And our understanding of the gut communities of many wild passerines were really limited when I started my PhD. And so this graph shows the accumulated number of studies on gut microbiomes of wild bird hosts. And as you can see, within the last 4 to 5 years, we are starting to increase the number of studies that are looking at these microbial communities. And with that, we are also starting to get a better understanding of what sort of factors drives these complex symbiont associations in bird hosts. And when we look at these factors, we can categorize this into a few groups. And for the studies have shown that many genetic and physiological factors influence these complex communities, and also ecological and behavioral factors. And finally, the environmental factors can also influence the assemble and the community composition of these bird microbiomes. So again, during my PhD, I wanted to understand how important and what magnitude each of different factors plays on structure in these microbial communities. So I had few specific questions that I would like to get answers for. So one of these is, are there any phylogenetic barriers on shaping gut microbiomes, specifically looking at whether there's a signal of phylosymbiosis in avian gut microbiomes. And here the phylosymbiosis mean that if the host evolutionary history is important for a structure of the community composition, then we would expect to see a good alignment between the host phylogeny and the microbial community compositions. And then I also wanted to look at what other ecological barriers influence avian gut microbiomes. So to do this, I used passerine birds from Papua New Guinea, multiple of them, multiple different species, and try to investigate their cloacal microbiomes. And to answer the first question, I used 37 passerine bird species from Papua New Guinea. And in this graph, it shows this ordination plot shows the microbial composition of different host species are used in the study. And the centers of each shapes are average microbiome composition of a species, and the colors indicate the family, bird family they are belonging to, and the arrow bars represent the individual variation in these microbial communities. And in order to test the phylosymbiosis, one of the prerequisites is these microbiomes depicting a species-specific signal. And I tested that, and I found there is a species specificity in these microbiomes of these tropical birds. And then I moved on to test for the phylosymbiosis. To do this, I generated a bird phylogeny using mitochondrial and nuclear genes sequences from these 37 bird species. And then I also generated a matrix of microbial similarity of the different species I'm using, and then try to align them together to see if we see an association between these two matrices. And when we tested it statistically, we do not find an association between these two, showing that there's no phylosymbiosis in avian microbiomes, indicating that there's a less importance of the host evolutionary history on structure in these complex communities. And then we wanted to answer why we might not see this signal for phylosymbiosis in birds, because this is a common signal that we see in other vertebrate classes, especially in mammals. So one main difference between birds and most of the mammals is that birds fly, and they have many morphological and physiological adaptations to sustain life in the air. One of these adaptations is when you compare similar sized birds and a non-flying mammal, we see that the bird have a shorter gut length compared to the mammal. And this shows, and also we see that the digestive tract length or the gut length of a bird is associated with the retention time, how long they keep their digester in the digestive tract. And taken together, this suggests that if you're a larger bird with a longer digestive tract, then you will retain your food longer time, so providing a more stable gut environment compared to a smaller bird with a shorter digestive tract. And with that, I hypothesize that if the flight adapted gut modifications are important for these community structures of gut microbiomes, then we would see larger birds with longer digestive tract to have more stable microbial communities compared to smaller birds. So I tested that. So in this graph, we have the body mass in the x-axis, which is a indicator or a proxy for the gut length. And in the y-axis, we have the measure of stability of gut microbiomes. And each dot is a species from the same data set where I had the 37 species. And here in the y-axis, the higher values means the gut communities are less stable in a species compared to low values where more stable gut microbial communities. And this is why, and it actually supports the hypothesis that larger birds would have more stable gut environments gut microbial communities compared to smaller bird species. And then I also wanted to test the importance of the host feeding guild on these microbial communities, where in this flower plot you can see that different species belonging to different feeding guilds have their own consortia of microbial symbionts. But when we look at individual species, we do see a high variation in these gut microbial communities. So here I'm only showing Melanocharis vestris gut microbiome composition. Again, in this ordination plot, we have different individuals' microbiomes, and the shapes represent which localities they were captured. And when we look at this ordination plot, we see that there are around 3 clustering of the gut microbial communities, and each cluster contain individuals from different localities. So one way we can explain this is that if 2 individuals from 2 different localities are feeding on a similar diet, then they will harbor a similar gut microbial composition. So then we took on to test this to see if actually natural variation in diet influence these gut microbial communities with the help of Katrina Sam and her group, where we collected captured birds from, again, in the lowlands of Papua New Guinea. And we collected the cloacal swab for the cloacal microbiome. And then we collected we induced regurgitation in these birds to collect what they have been eating at that time point. And then we collected another sample for the crop microbiome. And then we identified these the dietary items in the regurgitated samples using both visual identification and also metabarcoding. Before I go into more details on this, I will first show you the results from the diet part of this study. So we actually captured species belonging to 4 bird phyla, and then bird orders, bird orders, and then we also captured many individuals from different species within these bird orders. And when we look at so in this bar graph, I'm showing the major proportion of different dietary items we found in these individuals through the visual identification. And one thing that we can easily see is that there's a high variation in what individuals are eating in the same species. But when we try to compare this with the metabarcoding results, we still see that high individual variation within a species, but we don't see an agreement between our visual identification and metabarcoding. And when we look at it, there's only 12 individual samples where we find a perfect agreement between diet we identify through visual identification and metabarcoding identification. And this shows that the importance of, when we investigate these diets of natural diets of wild birds, we need to utilize multiple methods to encounter correct for inherent biases in these different methods we use. So now I will show you only the cloacal microbiomes of this study. So here are another ordination plot showing the cloacal microbiomes of the individuals we use. And here the colors depict the different orders of the birds that we used. And when we test statistically, we find that the microbiomes are, again, have a species specificity. Actually, here is the order level specificity. So different orders have their own consortia of microbiomes. And then we wanted to see the similarity in these microbiomes, whether it's actually governed by the similarity in the consumed diets of these hosts. So we generated a matrix of the cloacal microbiome similarity, and we try to align it with their consumed diet similarity, where we don't find an alignment, again, showing that whatever the diet the individuals are consuming is not directly associated with the complete similarity of their microbiomes. But then we also wanted to see if particular bacterial taxa in the gut community responds to different components of the diet differently in different bird orders. So to do this, we looked at proportions of different dietary items that we found in their diets and tried to align them with the relative abundance of different bacterial genera we find in their gut communities. And so here to give an example, and then we looked at Pearson correlations and then try to see if there are any significant associations between bacterial genera and dietary components. And here to give an example is the proportion of spiders that we found in their diets. And then each bar is a different host order. And then we look at the 30 most abundant bacterial genera. And we find that only in pigeons, we have a significant association with spiders in bacterial taxa. We did this for all the other major orders of insects that we found, or the invertebrates we found in their diets. And also we conducted a similar analysis for plant orders we found in their diets, where we find similar pattern, where we see that only certain bacterial taxa are associated with certain dietary items, and these associations differ between host orders. So in summary, what we find in my work is that the long-term associations between passerine hosts and their gut microbiomes differ from other vertebrates, particularly from mammals, where we don't see a strong influence of the host phylogeny on structuring these microbiomes. And this could be driven by a gradient of microbial stability associated with the gut lengths that are in birds that are associated with flight. And finally, we also find that only certain gut microbes are associated with particular consumed dietary items. So that not entire the diet composition of the diet influence the entire microbiome of an individual, but its particular associations between a microbe and a diet item is what drives the effect of diet on these complex microbiomes. With that, I would like to thank many people, and also the funding agencies and hosts that have been supporting me for the last few years. And yeah, with that, I'm done. Thank you. If you have any questions, and if you like to contact, my email is also in the bottom of this slide. Thanks.
Wonderful. Thank you very much, Kasun. And yes, we are welcoming questions from anyone from the audience, people who are on the Facebook watching us.